Ardan Patwardhan from EBI, who has many of us will be familiar with him from his work on developing the EM database and EMPIAR, obviously very important tools for the whole field. So he, Ardan, which I hope is going to appear in a moment. Yes, there he is. So Ardan did his PhD in Stockholm. He started in actually optical microscopy, laser scanning microscopy, and then eventually moved to London to Imperial College and moved into computational structural biology. And the last 10 years, he's been at EBI working in the EM, in the databases. And obviously that's had a very significant impact on the whole field. So Ardan's going to give us an update on the databases now. So if you can, Yuri, if you can close your video and, oh, got stuck. And Ardan, if you can share your screen. Good. Okay.
Okay.
Yep.
Yep. Thank you, Helen. Yeah, so I'm going to present what's new in EMDB and EMPIAR. And I'll start by giving you a brief introduction to the status of the archives, introduction to and the status of the archives. I'll then go on to highlight COVID-19 related structures in EMDB. Then go on to highlight some key meetings on archiving that have been held since the last spring symposium. And then finally, new and ongoing developments with EMPIAR and EMDB. cryo-EM data, the map that you get from cryo-EM experiments, that's deposited to the EMDB. If you have models built or fitted to the EM map, those get deposited to PDB. And for most major journals, deposition of maps and models to EMDB and PDB is mandatory. You can deposit, and we encourage the deposition of the raw data to EMPIAR. And EMPIAR also takes raw data as well as the maps from volume EM and soft X-ray tomography experiments. If you're doing correlative imaging, then the light microscopy from the correlative imaging experiments can go to the bioimage archive, which has been set up at EMBL EBI since 2019. And if you want the links to any of these archives, they're provided below here. So when it comes to EMDB, we had a major milestone this year. In February, we had we celebrated having more than 10,000 entries by having this poster. The poster kind of symbolizes what we do as EMDB, which is preserving EM structures in time. And for those of you who see this picture in the background, this is actually from one of the first 3D-EM Gordon conferences in 1985. So yeah, congratulations to the EM community for reaching this milestone. If you look at the status of EMDB and the maps released per year, you can see that we have steady growth, and last year we had more than 2,000 maps released. If you look at the distribution of the types of maps, most of them are from single particle experiments, and then from tomography experiments. And this year, despite all the shutdowns and so on, we have still had quite a healthy number of depositions. So we are standing at 1,371 depositions as of yesterday. And if you want to have a look at more statistics, you can go to the website provided below here. I'll go through a trends, single particle maps. The average resolution shown here by the red curve has been improving, and last year was down to 5.6 angstroms. The blue curve here shows the max resolution obtained each year. these are usually methodology-based works, and the last 4 years, they've all been based on looking at the apoferritin. Detector usage trends, Gatan K2 Summit has been the dominant detector in use. But if you look at the light blue curve here, this is the Falcon 3. You can see that it's been rising quickly. And last year, there were 313 maps by Falcon 3 compared to 114 in 2018. And just a note that I've switched off, I've just highlighted a few detector types in this graph. If you look at software usage trends, and again, I'll just highlight that I've switched off many of the other software packages, just wanting to highlight that
Ardan, we're losing sound. We're losing your sound. I'm not sure if there's something you can do.
Sorry, I'll just check if my line is still. Can you hear me now?
Yes, it's a bit variable though.
Okay, I'll try to be a little bit closer to the mic if that helps.
Thank you.
So the cryo-Spark usage has been increasing rapidly. This year, we already have 130 maps released, and this is to be compared with the 131 maps which were released in the whole of 2019. If you look at EMPIAR, we have steady growth in EMPIAR, and we have now 300, almost 300 entries, and totaling about 300 terabytes. And the lion's share of these entries are cryo-EM entries, but we are also getting more scanning electron microscopy entries and so on. So there has been a lot of interest in the COVID-19 structures, COVID-19 related structures. For those of you who are interested in getting more information about these structures, one good starting point is this link given below here, COVID-19 data portal. This actually has links to other bioinformatics resources as well. But if you go to the structures sublink, you will get links to both PDB as well as EMDB. Structures which are related to both SARS-CoV-2, but also to the previous SARS and so on. But I thought I'd give you an overview of what we currently have in the EMDB. So we essentially have 23 different EMDB entries, but these are related to 5 different works. 4 of them have been published and 1 of them is unpublished. If you look at the first 2 of these are of the SARS-CoV-2 spike glycoprotein in the open and closed states. And Agnel was showing you one of these structures. The third one is of the SARS-CoV-2 spike glycoprotein binding domain in complex with the angiotensin converting enzyme 2, so ACE2 receptor. And then we got 2 structures of the SARS-CoV-2 RNA-dependent RNA polymerase. What I've tried to do here is highlight structures in EMDB and PDB which actually have the SARS-CoV-2. So those are in bold. And the structures which are in green and bold, these are the ones where we actually have half maps. And I wanted to point out a couple of things here. So if you look at the first row in the table, you can see that we have 2 EMDB entries. So one corresponds to the open and one to the closed and one to the open state. But if you look at the models, we only have the model for the open state. But if you look at the paper, they actually have pictures and discussions about the closed state as well. So there, I would expect that there is a closed state model as well. If you look at the third structure here, which is of the glycoprotein binding domain, you have 2 EMDB entries which have got SARS-CoV-2 in them. So the 30,039 is of the ACE2 plus the SARS-CoV-2. But then they have done focus refinement of that to the EMD 30,036. So this one has got a high resolution SARS-CoV-2 structure. But we only have one PDB structure here, and this PDB structure is related to the one of the ACE2 plus the SARS-CoV-2, not the refined SARS-CoV-2. I'll get back to some of these entries in some of the later slides, but I also wanted to point out that if you look at all of these structures that we don't have EMPIAR data for any of them. And this goes back, so if you saw my appeal a couple of weeks ago on the mailing list to deposit data, so I would really like to appeal once again to the community to deposit their data to EMPIAR. If you have colleagues, to encourage them, if you're referees, to make sure that this actually gets deposited to the database. In terms of key meetings that we had last year, so I'd like to highlight two meetings. One is a volume EM community meeting that was held at the Wellcome Trust in 2019. So this meeting was, it's basically embryonic effort to strategically coordinate efforts to address community needs on equipment, software development, and archiving. And it's a little bit akin to the early meetings which were held in the cryo-EM community in the UK to try and organize things and to strategically organize things for cryo-EM. We hope to push the volume EM community sort of in the same direction and to get the same benefits of having this sort of strategic organization. Another important meeting was the WWPDB EM validation meeting, which was held in January of 2020. This was in a way a follow-up 2010 EM validation task force meeting. And the things that were discussed were, for example, improvements to validation reports, data that should be collected, for example, making half maps mandatory if used for validation, dealing with complex multi-map, multi-model depositions, which seem to be becoming more the norm. I'll now go through a few examples of some of the new developments that we have in the pipeline. And if you have been to our pages, you might be familiar with the visual analysis pages, which show sanity checking and validation information about the entries. And we have tried to improve these. We've added new features to the validation that we do. And these pages are now called the validation analysis pages. And if you check out our dev site, you can actually access these pages. So I'm going to show you a couple of, these are examples based on some of the COVID structures. So in this case, this COVID structure is of the spike glycoprotein in the open state. So you got one of the protomers sticking with the receptor binding domain sticking outwards, which is this one here. If you look at the atom inclusion by residue, so this is a plot that we've had previously. Blue here means that you have a high atom inclusion. Brownish red means that atom inclusion is poor. What we have now included is that we actually show an image where the atom inclusion, the coloring is by atom inclusion. So this picture makes it quite easy to see that this region here corresponds to the receptor binding domain of the protruding protomer. Now, we have been, people have raised concerns over the atom inclusion because atom inclusion is very sensitive to the recommended threshold level. And for this reason, we also now include the Q score, which was developed by Greg Pentilli. The Q score is not dependent on a threshold and is more robust in that sense. But even when we run the Q score, we get a similar type of pattern that the Q score for the receptor binding domain seems to be show a poorer fit with the between the map and model. And again, we also show an image of the Q score overlaid on the structure, and you can see that that is the receptor binding domain that has this poorer fit. If we also run a symmetry analysis on the structure now. The symmetry analysis shows the applied symmetry, which in this case is C1, because you have one of the protomers sticking out, so it doesn't have any symmetry applied. But if you run a symmetry check on it, it still finds a weak C3 symmetry, and the weakness is the fact that here you can see that the peak height is not very high. We're also adding more analysis based on the other data that's being provided. For example, if raw maps are provided or half maps are provided, then we also calculate images and power spectra based on this. This is another example of of validation analysis, again run on one of the COVID structures. And just to orient yourselves, so what you see here is this is a structure of the ACE2 receptor with the SARS-CoV-2 receptor binding domain attached to it. So what you see here in cyan, this is the ACE2 receptor. What you see here is the receptor binding domain, which is brownish. And as you can see, brownish, this corresponds to E and F, which are the in this diagram. And again, if you have issues with using atom inclusion and you look at the Q score, you can see something similar, that it is the receptor binding domain of SARS-CoV-2, which is the one which has a poorer map to model fit here. Another development that we have in the pipeline is that we have been data mining EMDB to develop a library of canonical map motifs of residues at different resolutions. And the idea is that this library of map motifs could be used for validation purposes, as well as for purposes of, for example, for model refinement. What we now have done is that we now have a validation tool which uses these canonical map motifs, and we now have, so we call it the Strudel Score validation tool, and this is now available as a Chimera X plugin. So this here shows you an example of the use of Strudel, again, to a SARS-CoV-2 structure. Just to guide you through this, what you see here is sort of the different residues. Green means that the model fits the map density. The motif for a motif for the model element that fits the map density well, whereas regions where you have the red here and you have suggestions for other map motifs which might fit better into the map density. And what's in this case, this band of residues from 894 to 930 have low correlation with library motifs. And what is being shown in the picture here is that in this case, you got a residue, a leucine residue modeled, but if you look at the map density, it fits much better with tryptophan motif at this resolution.
Ardan, I think it's time to start winding up.
Yes, okay, sorry. So just one more slide, which is that we've been redesigning our website. We did an online survey where we got more than 100 responses, and we did user interviews. So big thanks to everyone who took part in it. I won't go through any of the new functionality, but we have a much better search in progress, which will also have, for example, graphs based on so you can search and you have dynamically generated graphs. And with that, I just want to say thanks. Thanks to Gerard, who is the head of the Molecular and Cellular Structure Cluster. Thanks to all the members of the team, to our funders, current funders, previous funders, and also thanks to all of you for listening.
Right, thank you, Ardan. We've got a number of questions collected, so I'll pick a few of them. So there's several, first about EMPIAR. Alexander Payne has asked, what are some of the roadblocks keeping people from uploading? Is it necessary step to benchmark? Oh, okay, they're saying this is necessary for benchmarking processing improvement methods. So is there some blockage? Maybe I'll group the questions. So Chai Gopala Singham has also asked, is it still possible to deposit raw micrographs to EMPIAR after article publication and depositions? Yes. Sorry, one more in that group. Lou Gan has asked, how much storage in petabytes can EMPIAR support? So can you deal with all the published projects that are going on?
Yeah, so we have gotten quite a lot of funding from the UK government for raw storage. We can scale into the petabyte scale quite easily right now. In terms of the infrastructure, it is also now, we've done a lot of things in the background last year to sort of allow us to scale into the petabyte scale. So there are going to be teething problems. So if everyone starts uploading, we may have some issues, but I think we are able to cope with the data now.
Okay, maybe I'll just choose one more because we're a little bit late. So Ed Brignol has asked, are there efforts to flag and remediate discrepancies between information in the deposition and things that are in the paper? So he's pointing out that some entries indicate the use of one type of camera in the paper, whereas in the database deposition, there's a different camera mentioned. So I don't know if there's any remediation going on for things like that.
Yeah, so there's a continuous ongoing remediation process. So it often means that we get things reported to us, but we sometimes have to sort of block things, do things in blocks. So it might not be actioned immediately, but we are continuously remediating things. And if there are things that other people find that we have missed, then please do contact us and we'll try to address them as well.
Okay, thank you. I'm sorry, I think we're kind of a bit late now, so I think we'll have to stop. So thank you very much, Ardan.